So the third region of the small intestine, the ileum, is about 3.5 meters long. It's the site of water and electrolyte reabsorption, and the villi here are characteristically wider or broader than the jejunum, but the real defining feature is those Peyer's patches, which are continuous rows or contiguous rows of lymphoid follicles. These are located in the submucosa and extending up into the lamina propria. This image or cross-section of the ileum shows really nice examples of Peyer's patches. So here you can see them. Here we go. You can see them all lined up here in a nice row, and the follicles appear to be down here in the submucosa, and they have these dome-like areas which extend up here into the mucosal regions. So here are the follicles with the germinal centers down here, and then they have these dome-like areas. So in this, on the right side here, you see the scanning electron micrograph. These blue leaf-like, finger-like projections are the villi, and the colored green areas, these domes, are the dome-like portions of the Peyer's patches. These are the surfaces that are covered by those M cells that transport antigens across that barrier. That's shown nicely in this diagram, where you can see the M cells covering that dome-like area over the lymphoid follicles. And surrounding those dome-like areas are all of the villi. So the large intestine has several regions. It begins at the cecum, where the ileum empties into the large intestine. And at that area, there's also the appendix, which is a small finger-like extension that projects off the bottom area of the cecum. It proceeds upward into the regions of the colon, the ascending, transverse, descending, and sigmoid, and then to the rectum, and then out through the anal canal. The main function of the large intestine is to reabsorb any remaining water and electrolytes, and to eliminate undigested food and waste, which includes mostly fiber, bacteria, the bile salts, and desquamated cells from the entire GI tract. So what are the features of this region? In general, they call it a smooth surface. Now, that just means that there are no villi. So the mucosa has no villi and no plicae circularis. However, it does have crypts. So like the small intestine, there are crypts of Lieberkühn. However, here they are mostly mucus-secreting cells with intermixed with absorptive cells that are really just resorbing fluid and electrolytes. Another defining feature, other than the lack of villi and many straight tubular mucus-containing crypts, is the organization of the muscularis externa. So instead of having two continuous layers of smooth muscle, the inner circular and outer longitudinal, the outer longitudinal layer is organized into 3 bands called the tenia coli. And those are shown here in this kind of small thumbnail that I've got by the red arrows. The colon, or the large intestine in various regions, which is mostly colon, has both serosal and adventitial coverings, depending on where you are. So let's take a closer look at some of those regions. We'll start with the appendix. We've looked at this before when we looked at lymphoid tissue, and when we looked at some lymphoid vessels too, lymph vessels. So you can see the appendix has a large number of lymphatic follicles in the lamina propria, and some of them extending down into the submucosa. Just like the colon, there are no villi, but there are crypts. However, the crypts are very sparse here compared to the colon. It has a fairly thin muscularis externa with 2 uniform layers, no tennae coli. That's a feature of the colon area, not of the appendix. So onto the colon. The colon is mostly made of goblet cells with fewer enterocytes interspersed amongst them, closely spaced crypts, and again the tennae coli, which is the distinguishing feature here. This image shows the tennae coli very nicely, so you can see the inner circular layer here of the muscularis externa, and then the outer layer here, which is mostly just this band. The last region of the GI tract is the rectal anal junction leading into then the anal canal. So the main function here is to eliminate the feces or the undigested matter. The rectum has colonic glands, and the mucosal surface is highly infolded. Then, another one of those distinguishing features is that abrupt epithelial transition. In this case, you're going from the simple columnar in the rectum to the stratified squamous of the anal canal. You will also see the internal and external anal sphincters in the muscularis externa. So the internal anal sphincter will be smooth muscle, whereas the external anal sphincter will be skeletal muscle. And then further out, when you have passed through the anal canal and you're looking at the perianal region, you will see things like hair follicles and apocrine sweat glands, which also help you to ID this area. So, defining features, the change or abrupt epithelial transition, the anal sphincters, and the features of the perianal region, like hair follicles and apocrine sweat glands. This is a really nice longitudinal section through this rectal anal junction. So at the top of this image, you can see colonic glands in the rectum. And then here you see that abrupt transition in the epithelium. Here you have simple columnar, and then it changes right there into the stratified squamous of the anal canal. This is from our own slides selection, where you can also see these colonic glands of the rectum, and then you have that abrupt epithelial transition here, and this would be the anal canal down here. And that's it for today. We'll see you in lab.